Hello everyone, today I'll present our findings on functional evaluation and clinical classification of BRCA2 single nucleotide variants using saturation genome editing. I have nothing to disclose. So as we all know, the BRCA2 genes plays important roles for homologous recombination mediated repair of the DNA double-strand breaks as well as double replication forks. BRCA2 pathogenic variants, mostly protein truncated variants, can lead to increased lifetime risk of breast, ovarian, and prostate and pancreatic cancers. From a large-scale cohort study that our group has done, about 1% of breast cancer patients in the general population will carry BRCA2 pathogenic variants. In addition to protein truncated variants, there are also known established pathogenic missense variants. Shown here is a functionally critical domain called the DNA binding domain of BRCA2, where most pathogenic missense variants, those known missense variants, are found solely here in this domain. However, gaps still remain as many variants of ancestral significance, or VUS, have not been well understood, with more than 5,500 BRCA2 missense VUS currently reported in ClinVar. Most of these VUS are missense, intronic, or infirm indels. Many of them were identified through predisposition gene panel testing. It can cause a lot of problems for clinical management of these patients. It can be really challenging to assess their cancer risk for patients and their family members. Patients with VUS often lose their opportunity of getting enhanced screening, such as mammography or MR screening, or potential prophylactic surgery to prevent breast or ovarian cancers. They may also lose potential treatment options, such as platinum agent or FDA-approved targeted therapies like PARP inhibitors. To tackle the VUS challenge, our group has developed a BRCA2 DBD CRISPR-MAVE assay. We mainly focus on this functionally critical domain, as I previously mentioned, the DNA binding domain, from exon 15 to exon 26. Our target regions are basically divided by each exon, plus or minus 10 adjacent intronic regions. So each target region is approximately 150 base pair in length. Our system was basically adapted from the Nature 2018 paper, where we use HAP1 cells. So in these HAP1 cells, BRCA2 are essential for viability in these cells, meaning pathogenic variants of BRCA2 in these cells will be depleted, whereas benign variants in BRCA2 will eventually be enriched through this cell survival effect. And then we generate our amplicons, and using next generation sequencing, we sequence our genomic DNA as well as our variant library. And then we assign each variant a functional score and a functional category. With these functional data, we then can classify each variant using ACMG-EMP classification model. To analyze our BRCA2 MAVE data, our lab used a model called the Varco two-component Bayesian mixture model, developed by our collaborator at the Duke University. It is basically a linear discriminant analysis model with batch correction. It uses supervised machine learning to assign each variant a classification or a category. And this model uses nonsense and synonymous variance standards from our MAVE data as the training set. This model will iterate a normalized functional score from multiple replicated experiments, accounting for the variance between replicated experiments. The final output of this model consists of two parts. The first part is the posterior probabilities of pathogenicity with a prior probability of 0.2. So this prior of 0.2 was estimated based on the best performing in silico predictor alpha missense that approximately 20% of pathogenic variants were predicted in this BRCA2 DNA binding domain. And the second output of this model is the likelihood ratio estimates leading to 7 functional categories, which are equivalent to the evidence strengths recommended by the ClinGen ACMG guidelines. So as you can see, our model can clearly differentiate the nonsense and the synonymous variants, and they're fully separated in this functional score scale. And the results are also consistent in our exon-based analysis. After we apply this functional category, we can still see the same trend where almost all nonsense variants scored a pathogenic strong category, whereas almost all synonymous variants were assigned this benign strong category. So importantly, we wanted to validate the performance of our model. To do that, we use 4 independent known standard variant datasets. 2 are from the ClinVar and 1 from the ClinGen vSAT missense standards, which are the known pathogenic and known benign missense standards. And we also use a standardized HDR assay. So this assay was published early this year, and it was calibrated relative to the known benign and pathogenic missense standards. As you can see across all 4 standard data sets, our MAVE assay achieved high sensitivity and specificity for all the validation sets, suggesting that our MAVE assay performed very well relative to these standards. We also modeled the mutational hotspot of the evaluated variants onto the 3D structures of BRCA2 DBD subdomains. The darker the red, the higher proportion of pathogenic missense changes were observed in these residues. So as you can see, overall, most of these changes were observed at the secondary structures shown in these alpha helices and these beta sheets. And we also observed that those pathogenic missense changes were enriched at the helical and the OB1 subdomains. We then compare our MAVE results to 4 other published individual-based functional assays. As you can see, our MAVE data were able to differentiate the functionally abnormal variants versus functionally normal variants across all 4 individual assays with significant difference. In addition to the functional evaluation, we also wanted to know the clinical significance of our MAVE assay. To do that, we evaluated the risk of breast and ovarian cancers from our MAVE data. So the cases were collected from breast and ovarian cancer patients undergone genetic testing at Ambry Genetics, and the controls were from the public available NOMAD database version 4. So our MAVE missense pathogenic strong category confer high risk of both breast and ovarian cancers. And these odds ratios are also consistent across those pathogenic missense standard and the protein truncated variants. Importantly, we also found that the odds ratios for the P-strong categories are significantly different from our MAVE missense benign strong category or benign missense standard category. And finally, one wanted to formally classify these almost 7,000 BRCA2 SNVs using a ClinGen vSAT ACMG classification model. By using this model, we were able to classify more than 90% of SNVs in this hotspot, and with only about 8% remain as VUS. And for the missense category, we were able to classify more than 4,000 variants as either benign likely benign or pathogenic likely pathogenic, with only about 12% remain as VUS. Overall, we evaluate almost all single nucleotide variants in this mutational hotspot of BRCA2, and we assign them categories or evidence strengths based on the ClinGen ACMG guidelines. Importantly, our assay were validated against 4 different standards data sets, and we also achieve high sensitivity and specificity in these validations. Case control analysis showed that these pathogenic strong variants were indeed associated with elevated risk of breast and ovarian cancer. using a VSAT VSMG classification model, we were able to classify more than 90% of the SNVs at either pathogenic, likely pathogenic, or benign or likely benign. That means with our MAVE data, now carriers of pathogenic or likely pathogenic variants may qualify for enhanced screening, such as MRI or mammography. They may also potentially get treatment of breast or ovarian cancers with PARP inhibitors. So our findings can greatly enhance the risk assessment and clinical management of carriers from all racial backgrounds and for all BRCA2 associated cancers. And thank you. I'm open for any questions.
Thank you, Gilbert. That was fantastic. Questions from the floor? Okay. Perhaps I can ask. I'm going to show my ignorance here about knowing very little about cancer and anything to do with the BRCA gene. When you looked across the effects of mutations and you looked in patient populations, you had separated out by breast and ovarian cancer. Are there differences in the mutational patterns or location of mutation in the protein between the two types of cancer? I noticed there were differences in odd ratios and effect size, but was that due to the number of patients you had available to test, or penetrance, or anything?
So basically, the odds ratio analysis were pooled. We pooled like all cases observed with the P-strong categories from our MAVE data set. We didn't really observe a structural preference. Actually, the P-strong categories, like I mentioned, were enriched in the helical and the OB1 subdomains. I think that's the preferred location of these P-strong category variants.
Thank you. Question at the back?
Yeah. Hi, I'm Priyanka from UCSF. Fantastic talk. I have a question where, so in your data set, I saw that most synonymous mutations have a neutral effect. But I just want to ask that if you know that in cancer genes, are there any examples where synonymous mutations have had a dramatic effect? Because since we know that they can have a non-neutral effect. And secondly, in your data set, there was, I think, very little, but 1% of synonymous mutations, which did show a non-neutral effect. So did you see if they are true positives, and if it was, then why so?
Yeah, definitely. So these, not all synonymous variants in our assays had a benign category. We do observe that with potential, so we use this splicing predictor called Splice AI to rule out any potential splicing effect for those synonymous variants. And we know that a proportion of these synonymous SNVs will have some splicing effect, potential splicing effect. So that's how we evaluate those synonymous variants.
Hi, sorry, Arne, VUS Diagnostics. Great talk. Sorry if I missed it, but in your conclusion, you also mentioned this risk, the odds ratios for these missense variants, if I got it correctly, that you have identified. Did you also have a chance to compare the risk for BRCA2 and for breast and ovarian cancer for carriers of frames and nonsense variants versus the missense variants that you identify in your study? And is that the same, or is there any difference seen there?
So our assay, in the MAVE assay, we did not evaluate the frame shift variants, but we collected the cases from the Ambry Genetics, where we also did protein truncating variants, and the results are very similar to the MAVE nonsense variants, which were evaluated in our data set.
Okay, excellent. Thanks.
If there are no more questions, thank you very much. That was great.
Thank you.